Hey, welcome back to Ask a Creationist. I'm Todd Wood. I'm a creation scientist, and I'm here to answer your questions about creation, evolution, origins, science, religion, faith, Genesis, the Bible, whatever you got. I'm interested in hearing from you, and maybe we'll feature your question on a future episode. This week, we have a question that I have gotten several times, and I don't think there's any easy way to phrase this, so we'll just go with this. Human-monkey hybrid embryo things? What is this? What monstrosity are these scientists up to now? So maybe we should, sort of take this one step at a time, talk about what it is, and maybe then we'll discuss what it might mean, ethically speaking. So in the first place, we can dispense with the idea that this is somehow some sort of half-human, half-monkey creature that has been generated in some Chinese lab somewhere. At least that's not what's described in the current paper that's just been released. Instead, what we've seen here is a different process altogether. First, monkey embryos were generated by inseminating monkey eggs, and these are basically cynomolgus monkeys or crab-eating macaques, as they're called. So these monkey eggs were fertilized with monkey sperm to make monkey embryos. And then, after these monkey embryos were alive for 6 days, then human cells were injected into the embryo, so that the result is not a hybrid. So a hybrid would be a situation where you have a monkey egg that is fertilized by a human sperm, or a human egg fertilized by a monkey sperm, right? So that every cell is part human and part monkey. But that's not what happened. What happened instead is that the embryo itself is entirely monkey until day 6, when they injected these human cells into the monkey embryo to see if they could get the embryo to continue to grow and divide with the human cells present. The result then is called a chimera, and as I mentioned, this is distinct from a hybrid. So the chimera here referring to the old mythology of the animal that's composed of parts of a goat and a lion and a snake and so forth. And chimeras have been known for a long time. Scientists use the term chimera to describe all manner of types of things, and animal chimeras have been generated in the past, particularly rat-mouse chimeras and sheep-goat chimeras, both of which involve fusing of embryos of different species. This is not the first time that human cells have been put into an animal embryo. People have already been working on this sort of research, and it's been done with other species like pig and mouse, but this is the first time we've done it with a monkey, and it's probably the longest living of these chimeric embryos that have been generated. Basically, out of 132 chimeric embryos produced at day 6 of growth of the monkey embryo, by day 19, basically 2 weeks later, there were only 3 left alive. So this is a very successful research, but it's more successful than similar projects. Now you might be thinking, why would anyone want to do this? What kind of Frankenstein monstrosity mindset would try to wonder about what happens when you put human cells into monkey embryos? Well, the answer is actually kind of reasonable. Well, at least the motivation is kind of reasonable. And it's regenerative medicine. The idea here is how do we design effective and good treatments for conditions in which organs have been damaged, limbs or other structures have been damaged or worn out, and so forth, right? We're talking about things like amputations, we're talking about liver disease, kidney disease, maybe even, pancreas, replacing the islets of Langerhans in people who have diabetes so that they can stop having to take insulin all the time. We're talking about also spinal cord injuries. Is it possible to do something that would regenerate the spinal cord, that would restore movement to the otherwise immovable parts of the body? So the idea in regenerative medicine is instead of doing, prosthetics or inelegant transplants in which you end up having to take a bunch of immunosuppressant drugs for the rest of your life, what if you could just grow something new, right? What if you could grow your arm back, or grow a new liver, or grow a new heart, or grow a new lung, right? So it would be your own organ, it would be human, and it would be available for you, right? So the step people are trying to go through right now is to generate what are basically organs from other creatures that would be more suitable to transplant into human beings. The main target here is pigs, where pigs could produce organs. The organs of pigs are roughly the size of organs of human beings, and so we kill a lot of pigs for food, and the organs go to waste. Why not just transplant them into people? Well, to do that, we'd have to make the pig organs more suitable for the human host. And so there's this broad area of regenerative medicine research that is, frankly, well motivated. There's a very good motivation, and the idea of being able to do this is really phenomenal and amazing. And there are creatures out there. We know lizards, for example, or salamanders, when you pop their tails off, they will grow them back. So this isn't unheard of in the natural world. So the question is, how do we get human beings to do this? Okay, so that's why they did it, and that's what they did. Is this good? What are some ethical considerations to think about when we think about this research? The first thing that I remembered when I heard about this research is the natural occurrence of human chimeras already. So since the 50s, there have been dozens of cases of human chimeras that have been identified through various clinical examinations for one reason or another. And each one of these cases that have been identified are individuals that consist of cells derived from two different zygotes. Well, there's a couple of ways that it can happen, but the idea is that you have two different embryos in utero that fuse and become a single individual. And some of these individuals are otherwise normal. They look normal, they act normal, they experience life normally, nothing odd about them, except that some of their cells are different from others, right? So they're large portions of their bodies that are not genetically identical. But since they've had these cells since from embryonic stage, then the body recognizes the different cells as still the same body, right? So the body works fine. Other cases involved individuals with various abnormalities in their genitalia, in which it turned out that there had been the fusion of a male and a female embryo, which resulted in these abnormalities, and in fact, some true hermaphrodites. So in that sense, the idea that somehow chimeras of humans or chimeras in general would be some horrible, abhorrent thing against God's order of creation, I'm not sure that we can say that, since this does happen. It does happen naturally, and it seems to be, in some cases at least, harmless and only detectable under weird circumstances like genetic screenings or blood test screenings or something like that. Well, that said, are there actually problems with this research? And I had to dig into this quite a bit to answer all of my questions. And the first question I had was, well, where are these human cells coming from? The research paper describes these human pluripotent stem cells. Pluripotent here refers to cells that are capable of becoming any type of cell, a bone cell, a brain cell, a blood cell, heart cell, muscles, whatever. That's a pluripotent cell. And that would be the kind of cell you would need if you wanted to grow a new liver or you wanted to grow a new kidney or something like that, or even a new knee. That would be something that you would need to have. But the question is, where do they get it, right? Where do they get these cells? Well, they were derived from embryonic stem cell line H9. And where does that line come from? Well, that line came from research done in the late 90s that was done on leftover embryos from in vitro fertilization. So couples that had gone in to undergo IVF therapy, they got their pregnancy that they were looking for, and they had leftover embryos because, of course, many implantations just naturally do not take, and so pregnancy does not result, and so you have to do it multiple times with multiple embryos and hope that one of them implants. So there's always multiple embryos made, and then once you have the successful pregnancy, you have leftover embryos. What do you do with them? And so in this case, as they said in the paper, there was informed consent of the parents and then the Institutional Review Board approved the use of these embryos, and they were destroyed in order to harvest their stem cells. Now, notice here, the informed consent refers only to the parents, not to the embryo. The embryo didn't have any say in it. I find that problematic. Very problematic. Second problem I had with this research, the researchers in this present study with the human monkey chimeras compared their chimeric embryos to the normal embryos of each of the species, the cynomolgus monkeys embryos and human embryos. And these embryos were generated with the intention of doing this kind of research. In other words, they were created in order to be destroyed and studied. So human embryos were made in the course of this research in order to compare them to these chimeric embryos. I have a problem with that, big problem with that. To create an individual in order to destroy him or her for research purposes strikes me as, well, wicked, frankly. And then oddly enough, as I was digging around on this line H9, I discovered that there is a stipulation in the NIH stem cell database that says that H9 is not to be used for intermixing of human embryonic cells with an intact embryo, either human or non-human, which would seem to me to be exactly what this research is, and therefore would be a violation of the actual guidelines for using these cells. All right, well, that's another problem. So, and I don't know how they got around this. They claim that they had been reviewed by ethicists, although it seems pretty clear to me that H9 was not to be used for this type of research. But remember, this stuff happened in China, not in the United States. So there might be some other stipulations over there, although it seems pretty clear to me that this stipulation should be international, since H9 distribution is dependent on agreeing to these requirements. The paper that describes this work concludes the discussion, opens with this paragraph. There are significant ethical considerations involved in generating and studying human-animal chimeric embryos, particularly when non-human primates are involved. Not sure why non-human primates would make a big difference. I would think that using humans would be especially problematic. Different guidelines exist at the state, national, and international levels, and it is important for scientists, bioethicists, policymakers, and funding agencies to stay engaged in keeping these guidelines up to date with the relevant science, as well as for the welfare of society. Okay, so the science here, you've got to keep your ethical rules up to date with the science. That troubles me as well. I would think the ethical rules ought to be kept up to date with the ethics, right? And deciding what science is permissible and what is not. Strange wording there. For the studies presented here, extensive reviews of research plans and protocols were conducted in advance. Ethical consultations and reviews were performed both at the institutional level and via outreach to non-affiliated bioethicists with experience in state and national bioethics policies regarding these matters. This always strikes me, reading this, it strikes me as they are concerned about the politics and the rules, but not necessarily about right and wrong. This thorough and detailed process helped guide our experiments, which were focused entirely on ex vivo chimeric embryos. In other words, embryos that were never intended to be planted and grown up into an animal. So that was never their purpose. Their purpose was not to create organs for transplantation, which wouldn't even make sense because these monkeys are much smaller than humans. There's no way they could grow human organs. Human tissues, perhaps. But even then, it's way too early to know how to do that. Their main idea here was to just understand how chimeric embryos develop so that they could work on making their embryo development better. So for them, we didn't intend to make chimeric individual creatures of human and monkey origin. And furthermore, we limited our study to early stage chimeric embryo development. So at the stage of embryonic development when science is most uncertain about the full ethical status of the embryo. And that again strikes me as obviously problematic. If you don't know the status of a creature, then you don't kill it, right? If something might be human, it might be a human individual, then I ought not kill it for research purposes. I don't know, that seems like an obvious principle, right? And the principle would be simply, when in doubt, don't. But for some reason, our culture has it in their head that, since we don't know if these early human embryos are actual human individuals with human rights, then it's okay to go ahead and create and destroy them for research purposes. No, I don't accept that. I don't accept that whatsoever. And it doesn't even make any sense. If you don't know that these embryos are human individuals or not, you don't destroy them, because destroying them could be murder. And if you're destroying them for purposes of research, then that's the worst kind of murder. The response, of course, to this is that, well, we're helping people in wheelchairs get out of their wheelchairs, and we're helping people with neurodegenerative disorders, recover from their neurodegenerative disorders and so forth. And if you don't agree with me, then you hate these people. And that's just nonsense. We don't kill people to help other people. That's Ethics 101. You just don't do that. You don't murder people in order to help other people. And if there is a doubt about whether someone is really a person, you definitely don't murder them, because you might be guilty of murder. It seems obvious to me. So in the sense of these things being problematic as being, creatures with both human and animal cells, I'm not sure that's really so problematic. We use animal parts in human transplantation sometimes. For example, heart valves from animals that could be used in humans. And I don't have a problem with that. And the idea that you could use a pig to grow a human kidney, that sounds exciting to me. But the research that's necessary to get us to that point, very, 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 very difficult for me to imagine how that's done ethically. Yeah, when in doubt, don't. Very problematic. All right, well, thanks for listening. Please visit our website, coresci.org. There you will find links to our social media accounts. We're on Instagram, Twitter, and Facebook. You can follow us there, ask more questions. If you have problems with what I've said here, feel free to post them there. We'll consider what your objections might be. If you really enjoyed what we've done here, please consider a donation. We are a donation-driven ministry, and we would appreciate your support. Sign up for our newsletter as well. And thank you for listening. See you next time.